Hello and good morning. It's a pleasure for me to introduce Kalanit Grill-Spector. Kalanit Grill-Spector did her PhD in the Weizmann Institute of Science in Israel under the mentorship of Rafi Malach and Shimon Edelman. She did her postdoc studies with Nancy Kanwisher at MIT. She is a professor at Stanford University since 2001. Her research uses a multitude of neuroimaging methods in combination with computational modeling and behavioral measurements to elucidate the neural computations in the brain and the anatomical substrates that underlie visual perception. A significant part of her research is devoted to determine how these mechanisms develop from childhood to adulthood. She is well known for developing the method of MRI adaptation to study neural sensitivity in the human brain and for implementing innovative imaging techniques in her research, such as high-resolution fMRI, quantitative MRI, and encoding models. She has received several awards, including the HSFP Fellowship, Whitehall Foundation, Sloan Fellowship, and the Klingenstein Fellowship in Neuroscience. Her research is funded by the NSF and NIH. Today, she will tell us about her recent developmental research on brain growth and what else, facial recognition. Kalanit.
I would like to thank the OHBM Program Committee for inviting me to give this lecture. Today, I will tell you about exciting new research from my lab at Stanford University, looking at the relationship between brain growth and face recognition. I would like to highlight the contributions of my students and postdocs who perform this research, in particular, Golije Golari, Jesse Gomez, Vaideh Natu, Michael Barnett, and Brianna Jeska. So faces are important for our everyday social interactions because these are the key source of information we use to recognize other people, and it affects how we socially interact with them. Face recognition ability develops from childhood to adulthood. As shown in these three independent studies, adults are better at face recognition than school-aged children. Thus, face recognition is an ability that develops from childhood to adulthood. Face recognition is thought to rely on clustered regions in the fusiform gyrus that respond more strongly to faces compared to other objects, as shown for this individual participant. Specifically, in each subject's brain, you would find one region that is on the lateral fusiform gyrus, overlapping the mid-fusiform sulcus that we called M-fused faces, and another region that's about 1 to 1.5 centimeter more posterior that we call P-fused faces. These regions are often lumped together and referred to as the fusiform face area. Critically, responses in these regions are correlated with face perception. When you could look at a face and identify it, you get maximal activity in these regions. When you can tell that you're looking at a face, but you can't tell whose face it is, you get intermediate activity in these regions. And when you present a face and the subject misses this or doesn't perceive it, you get minimal activity in these regions. Critically, with fMRI, we can identify these regions in every single individual brain, even in our youngest 5 and 6-year-old subjects. So because we can identify these regions in each individual brain, and because activity in these regions is correlated with face perception, we believe that the face processing system in the brain is an excellent model system to investigate the development of specialization in the brain and its relationship to behavior. So today I will try to address two big picture questions. First, what brain mechanisms develop from childhood to adulthood? Second, how do these developments lead to enhanced face recognition behavior? In considering brain development, I will consider two competing developmental series, pruning and growth. Pruning has been suggested based on an analysis of post-mortem brains, especially in the macaques. And this series suggests that brain development is associated with pruning of unnecessary synapses and connections. For example, in a seminal paper by Pascal Rokik and colleagues, they showed that the number of synapses in primary visual cortex, or V1, decreases from infancy to adulthood. Pruning also has functional ramifications. Specifically, pruning is hypothesized to increase selectivity by decreasing responses to irrelevant stimuli. Another theory of brain development is called growth. This theory has also been based on analysis of postmortem brains, also in the macaque. However, this is in a different part of the macaque brain. It's called infratemporal cortex that is thought to be homologous to human ventral temporal cortex. Specifically, Elston and Fujita have shown that in IT cortex, the sizes of dendritic arbors, as well as the number of synapses, significantly increases in the macaque brain from infancy to adulthood. Growth also has functional predictions. Specifically, growth is hypothesized to increase responses and selectivity to the relevant stimuli. So we sought to address the predictions of developmental theories, both in terms of structure and function, using in vivo measurements in humans. As we're going to look at the structural and functional developments, we also will evaluate if there are any developmental behavioral consequence to these brain developments. So I want to tell you a little bit about our approach. We scan children between the ages of 5 and 12-year-olds and young adults between the ages of 22 to 25-year-olds. We train our children in a mock scanner to stay still in the scanner. Then we bring every subject to do some anatomical MRIs, and we do several functional MRI studies in each subject. In each subject, we also run a functional localizer in which subjects see faces as well as other stimuli, and we use that to localize high-level visual regions in the ventral stream. Importantly, we collect multiple runs, so we could use one set of data to localize a region and another set of data to extract signal and measure selectivity. We also conduct quantitative MRI scans in each participant, and then we bring participants to behavioral measurements outside the scanner to quantify their face recognition ability. This is a very intense data collection effort, and I'm going to show you data that includes more than 250 hours of data collection. I also want to tell you a little bit about our analysis approaches. We analyze our data in each participant's native brain space. We do not spatially smooth our data, and we use independent data to localize and evaluate both function and structure from different scans. And finally, we test and verify that there are no between age group differences in the measurement quality, motion, or signal to noise. So let's first consider the structural prediction of growth and pruning. Pruning predicts decreases in cortical tissue, and growth predicts increases in cortical tissue. Now, of course, with imaging techniques, we cannot make measurements at the cellular level. But what we're going to do is we're going to leverage new quantitative MRI sequences to test developmental changes in cortical tissue. Specifically, we're going to use a sequence that was developed by Aviv Metzer and Brian Wendell and colleagues at Stanford University. And this sequence removes the RF coil bias across participants, and it gives us a number with units that we can compare across individuals. So much like taking one's temperature, you get a number with units, then you can compare tissue properties across individuals. The number that I'm going to talk about today at length is T1 relaxation times. The units of T1 relaxation is in seconds. T1 relaxation time is inversely proportional to macromolecular tissue volume, or the volume of the tissue that doesn't contain water. That is, and it's also dependent on the composition of the tissue. Specifically, tissue with less macromolecules or more water would have higher relaxation time. For example, if your tissue is mostly within a ventricle, it's mostly filled with fluid, you have a long relaxation time of about 4 seconds. However, if you're looking in tissue that has many protons and lipids, such as myelinated white matter, you're going to measure a short relaxation time, for example, less than 1 second. So how can we use quantitative MRI to examine the predictions of pruning and growth? Remember, pruning predicts a reduction of tissue from childhood to adulthood. So if you have a proton that's perturbed by MR pulse in an adult environment, it has less tissue to exchange its energy with, and we expect that from childhood to adulthood, we have an increase in the T1 relaxation time. However, if we look at the prediction of growth, growth predicts that there's going to be an increase of tissue from childhood to adulthood. Therefore, a proton in an adult environment has more tissue to exchange its energy with and would have a shorter T1 relaxation time. Therefore, growth predicts a decrease in T1 relaxation time from childhood to adulthood. So we used fMRI to localize face and place-selective regions in each individual brain. I'm going to show you data today from p-fuse faces. We measure the T1 relaxation time in each individual. The left graph shows child data, the right graph shows adult data, and we see that there's a decrease in the mean T1 relaxation time from children to adults. This decrease in T1 relaxation time is also evident in the distribution of T1 values across voxels, as the distribution of adult T1 relaxation times is shifted to the left compared to children. So this decrease in T1 relaxation time is consistent with the prediction of the growth hypothesis. We also measured T1 relaxation time in collateral sulcus places, which is about 2 centimeters more medial to the face-selective regions. In the same participants, in contrast to the change in T1 properties, we do not find development in T1 properties from childhood to adulthood in an adjacent place-selective region. And this suggests that there are different trajectories of tissue development across face and place-selective cortex from childhood to adulthood. So does this structural development impact brain function? So what we did is we looked if there is a relationship between selectivity, or how much a region or a voxels preferred one stimulus compared to other stimuli relative to the tissue properties as measured by the T1 relaxation time. Each point in this graph represents an individual participant, where red indicates children, the circles indicate children, and the diamonds indicate adults. And what we find is that there is an inverse negative correlation between selectivity and mean T1 relaxation time. What that means is that participants with more tissue or lower T1 relaxation time had higher selectivity to faces in p-fuse faces. This relationship remains significant when we partialed out age. Interestingly, when we look at collateral sulcus places, where we do not find a change in T1 relaxation time across development, we find also no correlation between place selectivity and T1 properties in this region. So do these functional and structural developments affect recognition behavior? We assessed face recognition behavior using a test that was developed in Brad Duchesne's lab. It's called the Cambridge Face Recognition Test. And this is a test that uses, a variant of the test that uses child faces. So in this test, individuals are first trained to recognize 6 faces that are shown in different views. And after they've been trained, in each test trial, they're going to see a triad of faces, and they're asked to indicate which of these faces is one of the faces that they've learned. And as the test progresses, it becomes more difficult because faces are seen in new views and with noise. So now I'm going to show you the relationship between recognition memory performance as measured by the CFMT and mean T1 relaxation time. Again, we find a negative correlation between recognition performance and T1 relaxation time. That is, participants with lower T1 relaxation time did better at face recognition. And again, this relationship remains significant when we partial out age. Notably, this effect is specific. If you measure recognition performance versus T1 relaxation time in other parts of tissue, it is not correlated. And again, in collateral sulcus places where we do not see a development in T1 relaxation time, we don't see also a correlation with recognition even of places. So we showed that there is a tissue development that is consistent with growth, but many tissue compartments might contribute to this development. So we wanted to get a handle of a deeper understanding of what tissue compartments might be changing across development. One possibility is that there might be neurogenesis. Maybe the number of cells in the tissue is increasing from childhood to adulthood. One other interesting feature of our data is that if you look at the T1 relaxation properties of face and place-selective cortex in children, they're undifferentiated. However, when we look at the adult tissue, there's differences between T1 relaxation time in face and place-selective cortex. Specifically, face-selective cortex has lower T1 relaxation time compared to place-selective cortex, suggesting that it's more tissue dense. We asked, is a lower T1 relaxation in adults in face than place-selective cortex due to a higher number of cells or higher density of cells? So how can you measure the density of cells in the tissue? You will need to relate your data to histological measurements. Fortunately, the labs of Zillis and Emmons have described recently the cytoarchitecture of the fusiform gyrus. And in a separate paper in collaboration with their labs, we found that there is a direct relationship between functional regions in the ventral stream and cytoarchitecture. Specifically, our face-selective region, P-fused faces, seems to be within one cytoarchitectonic region of the fusiform gyrus called FG2, and our place-selective region seems to be within another cytoarchitectonic region in the ventral temporal cortex called FG3. So what we can do, because we're able to project our data back to histological slices, derive the gray level index, or GLI, which describes the cell density across layers. So we extracted the GLI from FG2, which is associated with face-selective cortex, and FG3, which is associated with place-selective cortex. However, contrary to our prediction, the density of neurons in FG2 is lower than in FG3. And this suggests that the decrease in T1 face-selective compared to place-selective cortex is not due to a higher density of cells. So if the number of neurons in this tissue is not growing, what aspects of the neuropil or the tissue surrounding the cell might be growing? One component of the neuropil that might be changing across development is myelin. Another quantitative measurement that we derived in our data is macromolecular tissue volume. The macromolecular tissue volume have been shown by Aviv Metzger to be directly correlated with myelination. And with our data, we found that there's a 13% increase in macromolecular tissue volume from childhood to adulthood. Therefore, we sought to simulate how much the myelin sheath needs to increase inside from childhood to adulthood in order to account for the 13% increase in macromolecular tissue volume. Our simulation used a variety of initial axon diameters as well as axon density in a voxel. However, we found that in order to account for this development in MTV, the myelin sheath would need to grow by a factor from 2 to 10-fold, resulting in axons that look very unrealistic compared to axon in the brain. So while myelin might contribute to this development, we think that myelin is not the only thing that is changing. And in fact, if we look back to the results of Elstone and Fujita, it's likely that increases in the dendritic arbors and number of synapses also contribute to this development that we measure in the T1 properties. Okay, so we found structural evidence for the growth hypothesis, and moreover, we found that these structural changes are also correlated with functional changes as measured by increased face selectivity and better face recognition performance. So while the tissue does not seem to be structural pruning, maybe we have evidence for functional pruning. So let's consider functional predictions of the pruning and growth hypotheses. The pruning hypothesis suggests that maybe perhaps responses in ventral temporal cortex are initially undifferentiated. Maybe each neuron and each voxel responds to a variety of stimulus categories, and what happens across development that as responses get pruned, you get more differentiated regions in children than adults. The Gross hypothesis makes a different functional prediction. It predicts that maybe in children, there might not be a lot of face-selective neurons, and maybe the size of face-selective regions would be small, and what happens across development will be an increase in the size of face-selective regions. So we sought to test these hypotheses by looking both at distributed responses across ventral temporal cortex to test if they become more differentiated across age, as well as measuring the size of functional regions in the ventral stream. So let me give you a sense of how these distributed patterns look in individual subjects. So the top shows distributed responses in an example child brain when they look at faces, objects, and places. And the bottom shows the distributed responses in ventral temporal cortex in an example adult when they look at faces, objects, and places. And just by looking at these patterns, you can see that they're rather similar across age group. For example, in both the child and the adult, we see this lateral medial gradient for the distributed responses of faces. So they look rather similar. So we quantified the representation similarity of these patterns by measuring the correlations between distributed responses across different categories. So the left shows the representational similarity matrix in average across our child participants, and the right shows the representational similarity of distributed activation in the adult participants. And you can see that these representational similarities are actually very similar across participants. They show a clustering of the three main domains of faces, objects, and places. We can also quantify how much information is in these distributed responses by using a decoding approach where you use an independent classifier to train from brain patterns what categories a subject is viewing. And we can see that both in children and adults, we can decode successfully what is the stimulus that they're looking at higher than 90% correct. And the pattern of classification is not different across children and adults. So this suggests that at least after age 7, the distributed responses to the domains of faces, objects, and places are rather stable and not changing across development. However, if we measure the sizes of functional regions in the ventral stream, we find a different pattern. Specifically, the number of face-selective voxels as well as the size of face-selective regions increase substantially from children, shown here in blue, to adults, shown here in red. And this development is particularly prominent in the right hemisphere, where we have about a doubling of the size of face-selective regions from childhood to adulthood. So these analyses of distributed patterns, as well as the sizes of functional regions, again provide evidence supporting the Gross hypothesis because we find specific increases in the sizes of face-selective regions. However, there might be multiple mechanisms that might explain increased selectivity across development. For example, pruning suggests that increased selectivity might be because of decreases to non-preferred stimuli. So imagine you're measuring a voxel in a face-selective region. Maybe it has low selectivity in a child brain because it does respond a little bit more to faces, but it responds quite significantly to many non-face stimuli. However, what might happen across development as you prune the responses to non-preferred stimulus, and you should get a decrease in the responsiveness to non-face stimuli. However, the Gross hypothesis suggests the opposite pattern, that what should happen across development is a specific increase in the responsiveness to the preferred stimulus, in this case, faces. So to measure the prediction of these hypotheses, we use one set of data to localize face-selective region and another set of data to extract responses in face-selective region in each of our child and adult participants. I'll show you an example from M-fused faces, but we get the same responses in other face-selective regions. The blue curve shows the responses in children, and the red curve shows the responses in adults. And you can see that there's not a difference in children and adults in responses to many non-face categories, such as bodies, cars, houses, and characters. However, we see a significant increase in the responsiveness to faces. And again, this is consistent with the predictions of the growth hypothesis. So we find increases to responses to the preferred stimuli rather than decreases to the responses to non-preferred stimuli. However, you might wonder, how does bigger face-selective regions and higher face selectivity give you better face recognition performance? So we hypothesized that another facet of functional development might be increased neural sensitivity to face identity. And this increased neural sensitivity to face identity might lead to better perceptual sensitivity and consequently better face recognition. So how can you measure neural sensitivity in face-selective cortex? One method that you can use to measure neural sensitivity is fMRI adaptation. Many groups have shown that the level of adaptation is dependent on the similarity among stimuli. So the more similar stimuli are, the higher that adaptation. So if you measure the response level to faces that vary with the similarity, you can measure the slope of this relationship, and the slope of the relationship would indicate neural sensitivity to changes in face identity. And what we sought to examine is whether there's a change in neural sensitivity, and specifically if the slope of this relationship is higher in adults than in children, indicating higher neural sensitivity. So to address this question, Vaidi Natu developed a really nice parametric fMRI adaptation experiment in which we manipulated the similarity among faces by morphing source faces to 6 different target faces. And by different weighting of the source and the target faces, we can vary the level of similarity in a parametric way. And to give you a sense of the stimuli, this is a example of 0 similarity faces. They're identical. This is 40% similarity, and this is 100% similarity. So we're going to measure in face-selective region the response level as a relationship to the dissimilarity measure. This is the average response from 19 children in M-fused faces, and we see that there is an increase in the response as the level of dissimilarity increases. In adults, as we measured before, we have an overall higher response to faces, but we also see that the slope of this relationship seems to be steeper. So what we did is that we measured in each individual child and each individual adult the slope of the signal versus face dissimilarity. So this is neural sensitivity, and we compared across age groups. And what we find in both P-fused faces and in M-fused faces is that children have lower neural dissimilarity to faces than adults. This is true when they look at adult faces and also when they look at child faces. Noticeably, there's also a hint of an age bias. So in M-fused faces, adults have higher neural sensitivity to adult faces compared to child faces. But in P-fused faces, children have higher neural sensitivity to child faces compared to adult faces. So does this increase in neural sensitivity affect perceptual sensitivity? We measured perceptual sensitivity to faces outside the scanner using a behavioral test. And then we examined if there's a relationship between perceptual discriminability and neural sensitivity. Each dot in the spot represents data from one subject, and the subjects are color-coded by age. And we see a significant and positive correlation between neural sensitivity in face-selective regions and perceptual discriminability. Notably, this relationship was significant. We didn't find it was specific and significant also when we partialed out age. Also, it was specific to face-selective regions as perceptual discriminability was not predicted by other regions that show adaptation to faces, such as object-selective cortex. Also, it wasn't predicted by other measurements of responses. For example, the overall level of response in face-selective region does not predict perceptual discriminability. So together, a series of empirical measurements shows that there support evidence for functional and structural growth. We see increases in the cortical tissue. We see increases in the size of face-selective region. We see increases in responses to preferred stimuli. And we also see increases in the neural sensitivity to face identity. So why might this growth occur? Faces are a particularly hard visual stimulus to recognize. After all faces have the same features and are arranged in the same configuration. So perhaps when you're a child, you're looking at a handful of faces, and they're rather different between maybe the faces of your siblings and your parents. However, as you grow, your social circle massively expands. And this requires you to discriminate and remember hundreds and maybe thousands of faces. And what we think that is happening, that this increase in neural resources, or more face-selective neurons, as well as an increase in neural tuning and sensitivity to face identity, enables you to be able to remember and distinguish among this multitude of faces. So to conclude, let's get back to our initial big picture questions. What brain mechanisms develop from childhood to adulthood? Our data shows that development of face perception involves both structural and functional growth rather than pruning of cortical tissue. Furthermore, our data suggests that pruning is not a ubiquitous mechanism that explains development, but instead different developmental mechanisms might happen across different parts of the brain and during different periods across development. How do these developments lead to enhanced face recognition behavior? We believe that cortical tissue growth in face-selective regions is coupled with both increased functional responses to faces and increased neural sensitivity to faces, consequently leading to better perceptual discrimination and recognition of faces. Our data has also several important implications. First of all, our results underscore the importance of obtaining anatomical, functional, and behavioral measurements in the same participants in order to understand brain mechanisms of development and also their impact on behavior. Furthermore, while my favorite part of the brain is visual cortex, the tools that I've described today are applicable not only for studying the visual system, but are also relevant to studying any cortical system and also across the lifespan more broadly. You might also ask, what might be the next big question? And one of the next big questions that we're exploring in the lab is, what is the role of experience in shaping brain development? And if you're interested in getting a sense of where our research is going, I'd like you to invite you to attend Jesse's poster later this afternoon. In this poster, he shows that there's differences in how children and adults look at or fixate in faces, and this difference in visual experience might affect both receptive fields as well as coverage in face-selective regions. So thank you for your attention.
Thank you, Kalanit, for this demonstration of functional and structural combined research on face-selective regions. Questions? Yeah?
I didn't know we were going to have an opportunity for questions. Thank you. That was absolutely fascinating. Your overall theory that the morphological changes in the brain are a result of growth in learning and exposure to faces is an interesting one, because not too many years ago, people might have grown in an environment where they were exposed to fewer than hundreds of faces, let alone learning the identity and discrimination among thousands of faces. Would it then be your hypothesis that, for example, on histological analysis, might one be able to go and detect differences in synaptic density or other morphological features in brains of people who grew up in an environment that's relatively reduced? And such people certainly would be available for study even now.
So this is a very interesting question, and basically you're asking if the size of your social circle might affect your tissue. And I, based on our finding, I would suggest that that would be probably true. I do still think that across many societies, the sizes of our social circles really expands with development. It's not only about it's also who we interact with and who's important for our life. So when you're a child, you're really, really interested about the close caretakers and your siblings and your family. And I think in many societies, as you grow up, you see a bigger circle in middle school and high school. And even if you're like maybe a hunter-gatherer tribe society and you have a smaller circle city, a social circle than in an urban society, I still do think that as you grow, you're going to expand. But I think that would be something that would be interesting to investigate across cultures that have variance as social circles.
Yeah, thank you very much. So you presented these two hypotheses sort of as competing, which has the great benefit that you get really clear predictions on what the neuroimaging results. My question is whether that's like the best way to characterize the phenomenon, given what Carla Schatz said yesterday about this sort of seesaw between the growth and pruning, and whether you think it's like just this like regional difference maybe in which these mechanisms are combined, or where do you see the place of pruning?
So I think, and also in relation to Carla's talk yesterday, I think based on the neural evidence, pruning is most likely to happen within the first 3 years of life. So in a human being, that would be between 0 and 3 years of age. It's very difficult to obtain fMRI measurements in babies because it's hard to give them instruction and hard to not make them move. I do think that some of the anatomical measurements that we've developed can be applicable to infant research because if you put a sleeping baby in the scanner, you could still get T1. So my prediction is that based on the, again, physiological research, that if there's going to be pruning, it's more likely to happen early in development, and it may be more massive in early sensory areas than in high-level visual areas. So that would be my prediction.
Thank you very much, Kalanit. I really enjoyed this. As you know, I've been advocating T1 as a measure of myelination since 2010. I have a specific question, though. Given that among the later myelinating fibers within the cortex, so the horizontal fibers comprising the bands of Baillet, do you think the increased selectivity that you're seeing here could be related to the recently demonstrated fact that most of those fibers are inhibitory rather than excitatory? Do you think that the myelination, the more efficient use of those fibers might actually have a role in providing greater selectivity for faces in the fusiform face area?
So this is a very good question. So I think it's likely that both excitatory and inhibitory inputs contribute to the shaping of tuning, and inhibition might be as important as excitation. One thing that we're actually looking in the lab quite actively right now is to looking at T1 as well as diffusivity across layers, and we do find some, it's not layers, it's about like across cortical depth. That would be a more accurate way to characterize it. And we find evidence that the bigger changes are happening close to the white matter, gray matter boundary, which would be very consistent with your prediction.
Thank you for the talk. I just had a three-part question, actually. So I know recent studies have shown that fusiform gyrus actually responds selectively to other modalities as well. So like lifelong bird watchers, if they see pictures of birds, it'll activate more than other objects. Lifetime mechanics, if they see cars, same thing. So I was wondering if you would think there would be a differential activation, like a separable activation for those kinds of things in people who are also experts in other fields, and how that kind of relates to expertise in pattern recognition overall for the fusiform gyrus. Second part of the question is, I know Stanislas Dehaene has showed some lateralization effects of the fusiform gyrus, showing that it's more left lateralized as you grow older with expertise. So I was wondering if you looked at that any. And the final part, I was wondering how you would think people with prosopagnosia, or at least some kind of face blindness, how they would show differential effects in the measures that you were showing. Okay, so you asked three questions. How does our research relate to expertise if we find evidence for lateralization, and what are the implications on our research on developmental prosopagnosia? So I'm going to answer them from last to first.
We think that these results have a big implication for developmental prosopagnosia, because one of the things we don't know is, this is an argument of the field, if it's developmental or if it's like you're born this way. And I think these T1 measurements would be a really good way to see if there are like some inherent tissue differences between a congenital or developmental prosopagnostic and typical adults. It's hard to obtain a big sample of studies, and in last conference in Vision Science Society, there's a big effort for the group of studying congenital prosopagnosia to start to do this. So I'm hoping that in the next maybe decade or so, we're going to accrue a large body of subjects, and that would really give us a way to address this. In terms of lateralization, we do see the evidence for lateralization for faces, and that the increase in face-selective regions is predominantly in the right hemisphere. We see a development in the left, but it's not as big. So if you actually look at children, they don't have lateralized face-selective regions. The size of face-selective region on the right and the left is the same, but it was developed and it increases more in the right, and leading to this lateralization in adults. If you're interested in learning more about lateralization, in Jesse's poster today, we actually looked at visual field representation in both face and word selective regions, and we do find, consistent with Stan's result, an increase, a shift in the left for words and a shift in the right for faces. Finally, in regards to expertise, even in experts, the responses to faces are higher than for their expert categories, and I think people argue whether it's the same neurons or different neurons within the fusiform gyrus. There's also some evidence from Marge Livingston's lab in monkeys that she's trained them on cartoon faces and on Tetris stimuli, that you'll get different domains but not in the same location. So we believe that there probably expertise might happen more broadly, and learning would happen more broadly in the fusiform gyrus, but it's likely not exactly the same voxels that are face selective.
Thank you.
There is a question here.
San, shoot. You mentioned that you may have different hypotheses in early life, and you're presenting on 7 to 12-year-old children, but what do you expect in adolescence?
Okay, so our data we've looked at 5 to 12-year-olds in the recent study. In older studies, we looked at 7 to 10-year-olds, and we looked also at adolescents that would be in between the ages of 12 to 16-year-olds. And there's some interesting finding. So the interesting finding is that face-selective regions and selectivity continues to develop into adolescence. However, place-selective regions and object-selective regions are already developed by then. So our data suggests that there is much more protected or much more prolonged development of face-selective regions compared to other domains, at least for objects and places in high-level visual cortex. What we haven't looked yet in more detail, and this is back to the previous question, maybe reading ability is thought to really change throughout the lifespan. If I would hedge a bet, this would be one domain that might be still massively developed through childhood and perhaps into adolescence.
And the last question over there.
Hi, thank you for your talk. So the question I had was regarding the change in the slope of the dissimilarity between the children and the adults. So I was thinking, could that instead be explained by changes in vasculature density? So there's a resting, say, density of capillary beds, which may increase as a function of age over time. So it may be that the effect itself hasn't really changed, but just the vessel dynamics have.
So this is a good question. How can hemodynamic differences across children and adults in general could account for any of our functional measurements with fMRI? And other people like Mariah Thompson and Gary Glover had looked at the HRF, basically the hemodynamic response function in children and adults, and at least in the age groups that we've looked at, over the age of 5, there doesn't seem to be big differences in the hemodynamic responses, suggesting that the vasculature is not that different. What we look in our term is looking how well the hemodynamic response function predicts our data, and we don't really find differences in the variance explained in the hemodynamic response function between children and adults. So I don't think that in our age groups the hemodynamic response function is changing. I think it's pretty mature in these regions. However, again, as you go in younger and younger age, I think you need to be much more more concerned. So if you look within the first year of life, I think we need to make, as a field, very precise measurement of hemodynamic response function in different areas because it might mature differently across the brain.
Thanks.
So, at the end of this session, I can only thank Kalanit for a brilliant talk and all the people who made a vivid discussion for their contributions. Thank you.